The overall goal of this procedure is to rapidly examine proteins bound to nascent DNA with accuracy, using BRDU labeling and chromatin precipitation.
This method can answer key questions in the replication, repair, and chromatin field, such as what proteins are bound to nascent DNA during DNA replication, and what repair proteins are bound to nascent DNA during replication stress. The main advantage of this technique is that it is a relatively quick method to detect proteins bound to nascent DNA in a highly quantitative manner.
To begin, set up cultures of 2 million NIH3T3 cells in 10 milliliters of media with either DMSO or inhibitors. Also, add BRDU to a final concentration of 20 micromolar. Once plated, incubate the cells for 60 minutes. After an hour, cross-link the intracellular proteins to the DNA by adding formaldehyde directly to the culture medium to a concentration of 1% by volume. Then, let the cells incubate at room temperature for 10 minutes with agitation. After 10 minutes, quench the reaction by adding glycine to the media to 125 millimolar and continue the incubation for 5 more minutes. Next, collect the cells in PBS and wash them twice with PBS using centrifugation. The pellet can be frozen at -80°C for storage if desired. Now, prepare the lysate from the cross-linked cells. Add 500 microliters of Fa140 buffer supplemented with a protease inhibitor cocktail to the pellet.